What makes you different to me? And how did it come to be that both of us look rather different from that mouse or tree? These were central questions for Darwin in his seminal work on the origin of species, and have continued to occupy biologists ever since. We now know all about genes and DNA, and have reconstructed phylogenetic trees of evolutionary history. Genetics and evolution can now tell us a lot about what differentiates individuals within a species, and indeed about what makes two species different from each other. While there have been some breathtaking breakthroughs and stunning insights along the way, one of the key developments between Darwin and today was made the slow way. Over 2 decades of painstaking work growing thousands of plants from dozens of species, Clausen, Keck, and Hisi compiled a treasure trove of information about how plants tick. Crucially, they elucidated to what extent different traits are controlled by genes and by the environment. Like any primary school science student knows, good experiments tend to change one thing, measure some dependent variables, and keep everything else the same. Normally, genes and the environment both vary a lot from individual to individual, so the trick to Clausen, Keck, and Hisi's experiments was separating out genetic and environmental variation. Humans wouldn't work for the experiments, as we can't clone many genetically identical humans, nor force them into identical environments. Plants, though, are ideally suited. They don't complain about boring lives in identical conditions, and we can easily take a cutting from one plant to start a new, genetically identical plant. In the original experiments, they planted some scruffy shrubs at different altitudes and examined how they developed, but that's old news and boring. So I'll present a reinvigorated, though hypothetical version of the same experiment using jacaranda trees. Jacarandas were introduced to Australia about 150 years ago, and have long been common on some university campuses. Let's choose 3, the University of Queensland, Sydney University, and the University of Melbourne. The jacarandas in each tend to be a little different, flowering earlier as you move north, and with some more purple-flowered and some bluer. We want to work out whether these changes to flowering time and colour are caused by the climate in those cities, or differing genotypes. Imagine that in my sagacious foresight, as an infant I planted lots of new jacaranda trees around the 3 campuses. I chose an existing tree in Brisbane and made 30 cuttings, planting 10 each in the 3 cities, and did the same from 1 tree in each university. By measuring atmospheric and soil parameters, I found 30 spots around each campus that were virtually identical for environmental factors, and planted my 90 cuttings accordingly. Now, I've come back 20 years later as an undergrad to examine my handiwork. Notwithstanding climate change, new construction, and nighttime festivals, let's assume the environmental conditions are still constant within each campus. Now, to tally up the results, I use my colorimeter to objectively quantify the average hue of the first 10 flowers to fall from each tree, and also note the date that this occurs. Here's what I found. The cuttings from all the 3 cities flower at about the same time here in Brisbane, even though the original trees in Sydney and Melbourne flower significantly later in the year. Likewise, flowering time is about constant at each campus. However, the ancestrally Brisbaneite trees have distinctly purpler flowers, with Sydney and Melbourne originating cuttings having bluer flowers, regardless of where the cuttings were grown. So, what does this mean? Well, flowering time must be controlled almost exclusively by environment, at least when comparing these closely related subspecies, as when placed in a common garden, all individuals had the same flowering time, despite differing genotypes. Colour, though, must be controlled by genes, as southern jacarandas, even once they were removed from their original climate, produced bluer flowers, and Brisbane jacarandas were purple wherever they were grown. While jacaranda flowers, and indeed Clausen, Keck, and Hisi's original species and traits, are rather trivial, this was the first time researchers had experimentally separated the effects of genes and environment, so what a big advance. Later scientists built on these techniques and ideas to eventually understand more complex and important traits like yield and drought tolerance in crops, and intelligence and disease proneness in humans. So this humble shrub really helped us understand how organisms are built and what makes them the same and different.